Hello and good afternoon, and welcome to the launch webinar for the BBSRC Proof of Concept programme. My name is Faye Shannon and I'm pleased to be speaking with you today along with my colleague James Phillips as we launch the first round of BBSRC Proof of Concept. Proof of Concept is BBSRC's flagship programme for bioscience translation and commercialisation, building on the work of the BBSRC Follow-on Fund and its more than 20-year track record of supporting bioscience innovation. The programme supports researchers to progress promising discoveries towards new products, services and processes with the potential to generate both economic and societal impact. The Proof of Concept programme is led by a team here at BBSRC, including myself, Ellie Tarkey, Vicky Gill and James Phillips. If you have any questions about the scope or strategy of the programme or would like to discuss whether a particular project might be suitable for Proof of Concept, please get in touch with us through the new programme mailbox, which is poc@bbsrc.ukri.org. Proof of Concept is delivered through the UKRI funding service, so if you have a technical question specifically about the funding service or submitting your application, we would encourage you to contact the funding service support team directly. The relevant contact details are shown here on the slide. We'll start today with a summary of the Proof of Concept programme and the support available. We'll then hear from some previous award holders who will share their experiences of the programme and how the funding has helped progress their research towards impact. Following the case studies, we'll provide a strategic overview of the proof of concept before taking you through the application process and what you'll need to consider when putting together an application. The Proof of Concept programme is BBSRC's flagship programme for supporting bioscience translation and commercialisation. Proof of Concept continues the work of the BBSRC Follow-on Fund, which has a long track record of supporting bioscience innovation. Over more than 20 years, the Follow-on Fund has invested over 80 million in more than 680 projects across the UK. In this latest round of Proof of Concept, we're seeking proposals with strong potential to progress bioscience research towards innovation and the development of new products, services or processes. This could include outputs such as spin-outs or licensable intellectual property with the potential to deliver substantive benefits to the UK. This reflects our wider vision as a public funder, which is to enable researchers to progress promising ideas from discovery towards impact, and in doing so, support successful and innovative bioscience community. This slide summarises the support available through the current round of Proof of Concept. We have a total of £3 million available. Projects can range from £100,000 all the way up to £800,000 at full economic costs, of which BBSRC will fund 80%. Standard grant costs are eligible and projects can run for up to 2 years. The earliest expected start date for awards for this round is July 2027. The closing date is 14th of October 2026 at 4 p.m. Please remember that your application will need to go through your organisation's internal approval process before it can be submitted to us. So we would strongly encourage you to allow sufficient time for this ahead of the deadline. Unfortunately, late applications cannot be accepted. We also plan to launch the next round of Proof of Concept in the new year. Before we move on, I just want to briefly highlight some recent changes to the programme. The main change is that the BBSRC Follow-on Fund has now been renamed BBSRC Proof of Concept. This forms part of a wider UKRI approach to simplify and better align translational funding across UKRI. Although the programme has a new name, the fundamental offer remains unchanged. The scope of the programme, the level of funding available, the assessment approach and the timing of our funding rounds all remain the same. We have also updated the programme mailbox, which is now poc@bbsrc.ukri.org, so please use this address for any future queries about the programme. In the previous round, we also introduced additional guidance around the involvement of international partners. The current opportunity has also been further aligned with the wider UKRI Proof of Concept approach. Overall, these changes are intended to provide greater consistency across UKRI while maintaining the established BBSRC programme and its support for bioscience translation and commercialisation. We're now going to hear from researchers involved in 2 projects supported through the Follow-on Fund, the programme which has now become Proof of Concept. Our first video looks back at an established project and the progress made through Follow-on Fund support, while our second video provides a forward look at a recently funded project that is just beginning its translational journey. Together, they provide some examples of the research supported through the programme and how proof of concept funding can help progress bioscience research towards translation and impact. I'll now hand over to our first case study.
Hello everybody, I'm Anthony Dodd and I'm based at the John Innes Centre in Norwich, and I'd like to tell you a little bit about our follow-on fund project that relates to the nutritional content of salads. This project is a collaboration between my group along with that of Martin Warren at the Quadram Institute, which is also on the Norwich Research Park, along with Jonathan Clark, who's the Head of Knowledge Exchange and Commercialisation at the John Innes Centre, and also an industrial partner, a company called Let Us Grow, which designs technology for indoor farms. And we've combined our different areas of expertise to develop this project. So my expertise is actually, the background is in circadian rhythms, but here I'm applying my knowledge of plant physiology. Martin Warren is an expert in the chemistry of vitamin B12, and Jack Farmer is a plant scientist by training who's interested in novel horticultural methods. So to provide some background and the motivation for this work, it relates to the deficiency of vitamin B12 in some people's diets. Vitamin B12 deficiency causes a variety of health problems, particularly problems with the development of the nervous system. And it's sometimes described as a form of hidden hunger because it can be undiagnosed and it's quite widespread. And that includes in the UK, where there's B12 insufficiency amongst a proportion of the population. And one of the things that's causing B12 insufficiency is an increasing shift to plant-based diets for environmental reasons or other cultural reasons. Plants don't contain vitamin B12, they don't make it, and we get vitamin B12 into our diets by eating animal products. Some people supplement their diet with vitamin B12 tablets. There's a couple of issues with those. Firstly, if they're not combined with a meal, the vitamin B12 won't be absorbed because it's only absorbed through the process of eating because of some factors that are secreted into the digestive system. And secondly, there's different isoforms of vitamin B12, some of which are active in humans and some of which are not, and the B12 tablets often contain a mixture of those things. So we reasoned that it might be possible to develop some alternative methods whereby a portion of salad is fortified with vitamin B12. So the aims within our project were to develop an approach that used aeroponic horticulture to fortify pea shoots, specifically we focused on, with vitamin B12. And this was a sort of experimental piece occurring both in my lab at the John Innes and through some experimental work at Let Us Grow's research and development facility. And this would be combined with a commercial piece by our industrial collaborator that related to the economics of the fortification process, so we would know, how much it cost and if it was ultimately successful, to help us to market the technology that we've developed. By bringing in the expertise of Martin Warren at the Quadram Institute, we were able to obtain accurate quantifications of the isoforms of vitamin B12 that are active in humans within the plant tissue. So aeroponic horticulture works, it's a little bit like hydroponics, except rather than the roots of the plant that is being cultivated, rather than the roots being immersed in a fluid with, for example, an ebb and flow supply of nutrient solution, instead the roots are suspended in a mist, which increases the availability of oxygen to the roots, seems to promote very substantial root growth. The technology that Let Us Grow have developed for aeroponics uses ultrasonic devices to generate a very, very fine aerosol. It also reduces the volume of nutrient solution that is needed in the system, which in the context of this project is really important because vitamin B12, the chemical, is incredibly expensive. It's more expensive than gold. So through a supplementation process into the nutrient tank, we wanted to have a method that would absolutely minimize the amount of vitamin B12 that we had to add to the system. In addition, during aeroponics, root development is slightly different and different cell layers form, and we think that promotes the uptake of B12 into the roots. So these are some of the outcomes from the project. This is just one figure from the paper that we've written about this. On this graph, there's a comparison of the accumulation of vitamin B12 in different parts of the pea shoots when they're at approximately the developmental stage shown with the cartoon on the left. And on this diagram, the broken line on the graph represents the recommended daily allowance of vitamin B12, and for each of the tissue types, the different bars represent the outcome when the pea shoots are supplemented with different concentrations of vitamin B12. And at least in the leaves, the tissue accumulated massively in excess of the recommended daily allowance. We calculated this costs about 3p to achieve this for one bag of salad. So the outcomes of the project are that we developed a set of processes to fortify pea shoots with above the RDA of vitamin B12, supplying at least 2 doses within a single salad bag sold within the supermarket. And we found that we did some experiments. It doesn't affect the shelf life, and it's also, by working with another lab at the Quadram Institute, we used simulated digestion to demonstrate that the vitamin B12 is bioaccessible during simulated human digestion, and so we think it would be absorbed during digestion. And the way that we're commercializing this work now is through 2 parts. The John Innes Centre and the Quadram are providing a certification mark for the product through our analytical services, so that a salad bag that is fortified using this method would have this certification mark on it, providing consumers with some confidence in the product that it contains this vitamin. And Let Us Grow is licensing the technology for achieving this to growers, including the protocols and equipment and so on. And I know that the approach isn't transgenic, so we think it would be accepted more widely. Where we are at the moment is that we expect the first license to be signed between Let Us Grow and a commercial grower during December. It's a grower that supplies one of the supermarkets. And in future, we think it could be quite interesting to try and stack this method with other forms of fortification or biofortification, such as increasing the iron content also through work of other labs at the John Innes Centre. And the other thing we're looking at is that we've been in discussion with the MRC about some funding for human trials of the product to just take it to the next step as well. The reasons why I think this project has worked, I mean, we've achieved all of this before the end of the grant, and I think it's worked well because we've got the right team with some very distinct and different areas of expertise, as well as the academic side. We've benefited a lot from the excellent Knowledge Exchange and Commercialisation Office at the John Innes Centre, and through having a business partner that has been very motivated to commercialise the work. And we also recruited an exceptional postdoc to the project. This really does make a difference sometimes, and they drove the project with really great enthusiasm and rigour.
Imagine a treatment for dairy cows that is fast-acting, effective, and designed to reduce reliance on conventional antibiotics. That is the ambition behind our project Pure Milk, which are peptide-based residue-free therapeutics for mastitis treatment in dairy cows. Pure Milk is a BBSRC follow-on fund project led from Queen's University Belfast with partners from the Moradon Research Institute Agriculture and Agri-Food Canada, and Raft Solutions UK. The project leads Professor Sharon Hughes, myself, Dr. Linda Oyama, and Dr. Nuno Silva, and the wider project team are turning antimicrobial peptide discovery into a practical veterinary innovation pathway for dairy cow health, milk safety, antimicrobial resistance stewardship, and sustainable food production. In Pure Milk, we are taking promising laboratory discoveries and generating the evidence needed to move them closer to veterinary use, industry licensing, and real-world impact. Our innovation is built around antimicrobial peptides, which are short, naturally inspired molecules with the potential to treat infection in a targeted way close to the site where mastitis occurs. Our aim is not to simply discover another active molecule, but to build the evidence package needed for translation. The problem we are tackling is bovine mastitis, an inflammatory infection of the udder and one of the most economically important diseases in dairy cattle. It affects cow welfare, milk production, milk quality, and has major consequences for farmers and supply chains. In the UK alone, mastitis costs the sector over £100 million each year. Conventional treatments are still largely antibiotics-based, and some require milk to be discarded during withdrawal periods when the animals are being treated. Our project Pure Milk asks a practical question. Can we treat mastitis effectively while reducing antibiotic residue, antibiotic resistance, and animal welfare concerns? The innovation of the Pure Milk project is based on antimicrobial peptides, or AMPs. These are short molecules that can kill or inhibit bacteria and other microorganisms, often through rapid interactions with the cell membranes. Our team at Queen's University Belfast has developed a library of more than 200 peptides through our earlier discovery work, funded through the BBSRC, the Invest NIU Northern Ireland, and other research agencies. From this pipeline, we have identified several promising antimicrobial peptide candidates. Our previous preclinical data, in partnership with our colleagues at Agriculture and Agri-Food Canada, show activity against a mastitis infection model in mice. We also show that the peptides are cleared from the milk, allowing the animal to return to production, while no detrimental effects to animal health were recorded. Our project, Pure Milk, is therefore a bridge between discovery and product pathway, allowing us to investigate a route to non-antibiotic veterinary therapeutics. Pure Milk is deliberately structured to allow technological advancement and an evidence base that would allow industry partners and regulators to translate the antimicrobial peptide candidates into solutions for mastitis. We will analyze how long peptide candidates are detectable in milk after intramammary administration. Second, we will develop and test peptide formulations and assess their efficacy in an experimentally induced mastitis model. Third, we will also move into naturally occurring clinical mastitis in dairy herds, where we can learn how treatment performs in the real world. Finally, we will build the commercial pathway, including the generation of intellectual property, engaging the market, and gathering market data, as well as having conversations about licensing to potential industry partners. This is where the follow-on fund model is especially powerful for our project. Pure Milk is not a single lab project. Queen's University Belfast brings the peptide discovery pipeline, microbiology, formulation technology and coordination, as well as the translational leadership, while the Moradin Research Institute brings expertise in ruminant infection models and controlled animal studies. Agriculture and Agri-Food Canada, AAFC, will support the residue study in lactating cows, generating evidence essential for dairy therapeutics. And finally, RAF Solutions will bring the farm-facing veterinary and commercial networks needed to test our advanced peptides in naturally occurring clinical mastitis and understand adoption in the real world. By the end of our project Pure Milk, success will not simply be a scientific publication. Our evidence will provide robust residue, efficacy, microbiology, and safety relevant data that show how the peptides behave in milk after treatment, efficacy data from controlled and real-world mastitis studies, and microbiology and molecular data that help us understand treatment response. We also want the information needed for patent filing, regulatory discussion, veterinary medicines directorate engagement, and future commercial licensing. Most importantly, we will work towards a practical treatment concept aligned with veterinary and farmer needs. The potential impact of this work reaches well beyond a single treatment. If successful, this work could contribute to better animal welfare by giving veterinarians more treatment options for a painful and common disease. It could reduce milk losses associated with withdrawal periods, supporting farm economics and food security. It could help address antimicrobial resistance by creating new non-antibiotic approaches for a major livestock disease. And it could contribute to sustainability because healthier cows produce more efficiently and waste less milk. Over the next 24 months, Pure Milk will focus on de-risking the technology. In the early stages, we will align protocols, prepare formulations, begin residue work, and ensure that partners have the right materials and documentation. As the project progresses, we will generate data from the induced mastitis model, integrated microbiology and animal health outputs, and refine the evidence package. In the later stages, the naturally occurring clinical mastitis study will provide crucial insight into real-world performance, while we prepare data for patenting, regulatory engagement, publication, and licensing discussions. Pure Milk is therefore about turning a strong scientific discovery into a credible innovation pathway. With BBSRC support, we can generate the data needed to understand residue behavior, therapeutic efficacy, formulation readiness, and real-world veterinary use. We can work with partners who understand dairy cows, farms, veterinary practice, and commercial translation. Our long-term goal and vision is simple: healthier cows, safer milk, fewer losses, and new tools to help reduce dependence on conventional antibiotics. Thank you.
We'll now continue with information about how to apply and the core scope. The BBSRC Proof of Concept program is our flagship translational program and an integral part of how we invest in research and innovation to advance the frontiers of biology. The program provides dedicated funding for BBSRC award holders to progress their ideas towards products, services and processes with the potential to deliver impact. The program supports early-stage projects that de-risk innovative ideas and later-stage projects with significant technical milestones. Successful applications will be within the remit of BBSRC and will demonstrate scientific and technical merit, economic and societal impact, and a strong potential for added value. They will also have a clear approach to managing any IP.
Applications are made using TFS and will be accepted until 4 p.m. on the date shown.
The project lead is responsible for completing the application process, and this includes making sure it is submitted before the deadline. The application can only be submitted by the lead research organisation. To start your proposal, visit the Funding Finder webpage and click the blue button at the top that says Start Application. This will prompt you to log into TFS or to set up an account, after which you can access the application form. Your proposal must meet the scheme requirements to be accepted for assessment, and this includes eligibility for BBSRC funding. The programme uses the standard UKRI eligibility rules for Research Council grants. This means you must be employed at an eligible institution and at a suitable level or higher. If you're unsure if you or your institution is eligible, then please inquire using the eligibility mailbox. The programme aims to secure economic and societal impact with the UK as the primary beneficiary. Funding is awarded through commercialisation grants to UK research organisations that meet UKRI eligibility requirements. Official development assistance projects are not in scope. These are projects that would primarily aim to promote or specifically target economic development or welfare outcomes in ODA-eligible countries and territories. Proposals to prevent disease outbreaks in the UK are in scope, and for these projects it may be appropriate to include activities outside of the UK where there is a credible pathway for movement. Proposals that seek to access export or investment opportunities for the UK may also be in scope, and this includes securing access to international supply chains where the UK is the primary beneficiary. The BBSRC Proof of Concept programme enables researchers to progress previously supported work towards impact. Proposals must draw upon current or previous BBSRC investments and enable research outputs to be further developed into practical application. Eligible previous BBSRC awards include research grants, studentships, fellowships, and work funded by BBSRC through both delegated and block awards, such as seed awards, IAs, IKCs, and networks. If you're unclear, please email the POC mailbox to check. Your award will also need to be within the standard scientific remit of BBSRC. BBSRC funds bioscience with a focus upon plants, microbes, animals, humans, and tools and technologies that underpin biological research. This includes work on all scales, from landscapes to molecules. This slide has some common queries about our remit. BBSRC does fund research relating to humans, but does not fund research on specific human diseases or abnormal conditions. In that case, MRC would likely be the relevant funder. To be within BBSRC's remit, the research should be upon the biological mechanisms relevant to the system. For proposals with a focus on technology development, we will often ask if the technologies are platforms rather than clinical or medical interventions. Other areas that commonly lead to questions include animal diseases, where studies primarily focused on animal models of human disease and toxicity are not in BBSRC remit. Research in wild species will tend to be within the remit of NERC, except when they are pests in an agricultural setting or pollinators of agricultural crops, or where they act as a model. For non-terrestrial systems, BBSRC will fund projects related to aquaculture and model species such as zebrafish, but often projects related to marine biology will be supported by NERC. Finally, BBSRC will fund research related to managed land, but only where this relates to agriculture, horticulture, and aquaculture. Project partners are not a requirement. However, they are encouraged as they strengthen most proposals. Partners can be from businesses and other user groups, but they must add value and should be connected to the aims of the project. If your project does not have any partners, then we would still advise you to clearly explain how users will be engaged. Partners are required to provide a letter of support to confirm that a collaboration agreement will be put in place. Please note that the standard guidance for subcontractors on Research Council grants also applies to this programme. BBSRC Proof of Concept Awards provide flexible options for partnering to better enable clear pathways towards commercialization. Project partners may access intellectual property by making a cash or in-kind contribution. Partners may secure a first right of refusal to negotiate access to foreground IP at the end of the project by making a contribution of 10% of the total project costs. Alternatively, partners seeking to pre-negotiate access to foreground IP must contribute a minimum of 50% of the project costs. A calculator is available on the Funding Finder webpage to assist you in working through the finances. Empty shell companies with no investment, staff or premises, which are formed for the sole purpose of commercializing foreground IP generated during the project, are not required to contribute. Applicants are advised against the direct assignment of foreground IP to a newly formed spin-out company. Instead, option agreements should be utilized until the spin-out has secured enough funding to drive the translation of foreground IP, when a substantive agreement should be executed. Your chosen approach for your foreground IP should be clearly justified in the application. Project partners are encouraged to provide clear justification for the value of any cash or in-kind contributions in their letter of support. This should include how those contributions are relevant to the project. Let's move on from requirements to the assessment of the application. Each proposal will be assessed in terms of its scientific and technical merit, societal impact, economic impact, and added value. The full descriptions of each of these criteria are provided on the Funding Finder page, and this is the same text that we will provide to the assessment panel to brief them on how to approach the assessment. This slide has an overview of the questions you will be required to complete when you are writing your proposal. While I'm not going to read out the text on the web page for each question, the point that I would like to clarify is that you will find a full description of each question on the web page for the opportunity. This guidance includes a description of what the assessors are looking for, and this text is the exact same briefing that we will provide to the assessment panel, so you have complete sight of how we will be asking panel members to approach assessing each question. Please do bear this in mind when writing your responses. These are the core questions in the set that will form the case for support, and I'd like to talk about them in a little bit more detail. The background section is a space to tell the story of how you've progressed to the point where translational funding is timely. Applicants are asked to describe the opportunity and market they are seeking to progress towards to develop or to deploy a new or improved product, process, service, or technology. When describing the route to market, you should consider how the proposed project will progress the innovation towards market and application. The question about intellectual property management and communication is an opportunity to describe how your project will progress the innovation towards translation. This includes your plans to manage IP, any dissemination, and if necessary, the potential to access further investment. In the section about wider benefits, you can describe the impact beyond the immediate commercial opportunity, including the potential societal, environmental, and economic benefits of the proposed approach. Finally, we ask whether there are any ethical considerations associated with the outputs of the project, including those that are beyond formal regulatory and legal frameworks. So I'm going to start to wrap things up now with some general advice for applicants based on how proposals have performed in the past. When writing your proposal, we would advise you to take care to ensure that the proposal demonstrates a clear understanding of what the customer or user needs and how those needs will be addressed appropriately in the FOF project. It's important to focus on the most appropriate opportunity for commercialization and to have a clear development plan. Any risks should be addressed directly with a clear description of any potential impacts and what steps are proposed as mitigation. Management of any intellectual property should be clearly described, including how any foreground IP will be managed. It's also important to have purposeful and clear project partner contributions or user engagement that directly relates to the aims of the project. We would also advise that there are restrictions upon how public funds for research and innovation can be used. Proposals with costs that are ineligible for public funding cannot be awarded and so will not be accepted for review. Please work with your local research administration office or your technology transfer office within your institution to seek advice on allowable costs and roles. Recent faults for proposals have included projects that are focused upon ODA research, which is out of scope, including research organisations that cannot receive UKRI funding, including businesses in roles for which they are not eligible, team members associated with incorrect roles for which they are ineligible, proposals that are not within BBSRC's bioscience remit, and project partner contributions that do not meet the requirements of the IP framework as described earlier. Proposals with these types of problems cannot be funded, regardless of how they might potentially score. As a consequence, faulty proposals are declined without proceeding to panel review. I'd now like to briefly share some other opportunities for translational activities. There are a variety of other funding routes for partnering, including BBSRC's Industrial Partnership Awards and Link Awards. These schemes are both embedded within our responsive mode and support collaborative R&D. They are a fantastic opportunity to advance partnerships and can be used prior to a Proof of Concept award. The iCure programme from Innovate UK is a pre-accelerator that supports teams led by academic researchers to validate their business ideas. iCure has developed a strong track record in supporting researchers to move their science from lab to market and is highly recommended for researchers considering a proof of concept application. Strategic support is also available for venture. And as an example, the BioBic program, which is a partnership with STFC, supports early-stage biotech startups that are built on leading bioscience research to bridge the gap between science and market to achieve their full commercial potential. Startups participating in BioBic receive a package of support, including funding, access to incubator space, and coaching, all to accelerate technology development and attract private investment. The program is just one example of how we're working to develop the bioinnovation ecosystem. We support collaboration through forums that bring together communities of researchers, innovators, businesses, and entrepreneurs to connect with each other, to share ideas, expertise, and to generate new biosolutions. These interactions stimulate impact, and joining our networks, hubs, and centres is a genuine opportunity to connect and to find new partners. UKRI supports Confer, which is the UK's smart matching tool to connect universities and businesses that are seeking collaborators. The tool can be accessed at confer.online. If you would like to be the first to find out about new opportunities from BBSRC that relate to translation and commercialization, then please subscribe to our bulletin. It's a newsletter that comes out regularly, which shares the latest opportunities that are of interest to people who are engaged in translational activities. And finally, if you would like to find out more about the type of awards that BBSRC and other UKRI councils have made, then all publicly available awards can be found on our Gateway to Research portal. Thank you so much for your attention. I hope that you found the presentation today useful. And thank you again to our guest speakers for showing us how much can be achieved through commercialization grant funding. The webinar continues now with our live Q&A session. Otherwise, feel free to get in touch using the contact details on the screen. Thank you.